So I'd like to begin by thanking Vijay for inviting me to speak here and tell you or give you the biologist's view of what is exciting about morphogenesis. I'll try and keep this simple, and I'd like to also ask you to interrupt me whenever you feel you're not understanding something. And Vijay and I will try and sort of do the physics and biology side by side, although I'd say that today's lecture is a bit of a tester to see whether we're sort of reaching out adequately or not. So you let us know what you think. Okay. So since there are many of you from outside India, I'd just like to point to this picture, and it's a picture of a peacock that's made by my students by hand using 2 fingers and let's say something that looks like powdered sand, and it was done on a working bench in my lab. And what you will see is that it is, of course, a peacock. You can tell from its shape that it is a peacock, but obviously it's also a hybrid peacock, because if you look at every structure here, it's patterned a little bit differently. We all know peacock's feathers don't exactly look like that. But I think what you'll also see is that, and I hope this will become evident in the slides over the next few talks, is that each of these feathers displays some sort of spatial pattern that biologists like me are interested in. You could call them patterns that have some sort of radial polarity that emanates from the center outwards, patterns that form strict boundaries and distinguish, let's say, cells or something else on 2 sides of it, and so on and so forth. And so really what this to me nicely reflects is the diversity of patterns that we see in living systems. And what we will try and look at during the course of these lectures is to understand how you build living structures. And this is really what morphogenesis is all about. How do you build those patterns? So if you look at morphogenesis, the word is derived from the Greek morpho, meaning shape, and genesis, meaning creation. So literally, when we speak of morphogenesis, we mean the creation of shape. Architectural firms today, and I was just looking in Bangalore, call themselves morphogenesis, but anyway, that's nice. So essentially, as biologists, we refer to morphogenesis as including the processes by which tissues, organs, and organisms acquire the shape or form that's critical to their function. And so you will quickly realize, looking at the number of animals around you, that not all creatures have the same form. So you'll have cacti that look like this, peacock whose feathers are patterned like this, butterflies, zebras, giraffes, and so on. Their body shapes are different, and the patterns associated with parts of their bodies are also different. And Darwin recognized many, many years ago that that what was fascinating was that from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. He, of course, spoke about the grandeur of evolution that generates animals that are so varied in their forms, if you like. And I think in one of the lectures, Professor Vijaykumar will try and elaborate on the evolution of form across between different organisms. And so really the timeline that Darwin was talking about was the evolutionary timeline, which is to say that starting from animals that are relatively simple, you sort of evolved animals that come in a wide variety of shapes and sizes and principles that govern their formation. What I'm going to speak about for most part is this timeline, which is the developmental timeline, which is how you make from so simple a beginning, which is the single cell, something quite beautiful and complex, which is the embryo or the animal. And what you see here are the stages in the development of a human embryo, right? You will see days, weeks, and so on. So this is quite a prolonged process to get to the final form of a human being. And this question of understanding how from a single cell you create an organism that has a shape that's quite unlike what a single cell might on its own be capable of producing is what will be the focus of my lectures. Joseph Needham, who was a biochemist at Cambridge, recognized many years ago that actually the central problem of biology is the form problem. And he said something interesting, which is that the hierarchy of relations from the molecular structure of carbon to the equilibrium of the species and the ecological whole will perhaps be the leading idea of the future. So I'm hoping that this meeting will somehow cover all aspects of that. And Needham wrote two very influential works during his life, Chemical Embryogenesis and Order and Life. The second one is a lecture. But he probably became better known for his interests in history and Chinese history and comparing Eastern and Western schools of scientific thought. So although this was called his magnum opus many, many years ago, he sort of bettered himself when it came to history. So morphogenesis is something that us biologists are interested in because we're interested in asking how you make complex and diversely patterned tissues from simple cells. And we're interested in this because it's fascinating by itself, and we want to try and ask how structures are built inside an animal, whether there are any fundamental laws that the parts that make up the animal follow in building these structures. So in a way, we're interested in understanding how patterns and structures are established. And many biologists, you'll, I'm sure, have heard, are also obsessed with how the function of genes contributes to the elaboration of this pattern. But there's also another class of scientists who are interested in morphogenesis, and these are people who call themselves tissue engineers, and they're interested in applying laws that one might get from studying animals in the lab to try and build artificial structures in a dish, right? So the question that motivates them is whether one can create three-dimensional tissues or animals in a dish. And if time permits, we'll spend a little time talking about what can you make in a dish and what does that actually tell us about how tissues are patterned. But if I go back now to the first steps in the making of a human, right? This is just up to the first 23 days, and you know there are 9 months. It takes 9 months to make a human. You will see a few general principles, right? One is that starting from the zygote, which is the single cell formed after fertilization of the egg and the sperm, you have what you call cleavage or divisions that increases the number of cells that make up this embryo, right? What you will also see is that with time, rather than just having one type of cell, the yellow cell, you start to have yellow cells and red cells. For simplicity's sake, I'll say cells on the inside become red and cells on the outside stay yellow, right? And then you start to build some sort of geometric complexity, which is that you make a somewhat symmetric looking structure now asymmetric, so that the red cells are skewed to one side of this circular structure. Then you start building cavities and linings and so on, right? So if you think of morphogenesis, it's associated with an increase in the number of cells starting from that single cell. It's associated with an increase in the number of types of cells. And in order for you to get these complex geometries, the cells also have to sort of move around from their initial positions to some later position. And what is visible is that it's geometrically more complex. So a question, and this is true whether you look at the embryo of a human or the stages in the making of a frog, which many of you would have seen in a pond, or at least many of you would have seen tadpoles in a pond becoming frogs and eggs of frogs in a pond and so on. So some aspects of morphogenesis, therefore, appear to be conserved. And I just want to point out here that I'm talking about animals that have lots of cells in them. I'll come back to animals that live their life as single cells. So so you can then say a frog looks different from a human, looks different from some other animal. So do we have to worry about morphogenesis as many problems, or one problem? Is there one solution or many solutions? And this is a sort of infamous picture from Ernst Haeckel's book called Anthropogeny, which has in each column some of the embryonic stages of a fish, a salamander, a turtle, a chick, a pig, a cow, a dog, and human. And one of the arguments that Haeckel made, which has been very controversial, was that the embryogenesis of an advanced animal, let's say a human, resembles the adult stages of a less advanced animal, for example, a chick, right? So he stated that ontogeny recapitulates phylogeny. This has been a controversial idea. We won't get there today, but what I want you to see is that at face value, some aspects of the embryogenesis of animals as diverse as fish and men look similar. So what people have actually begun to look at is, are there commonalities in the way different animals develop? And again, I'm not going to go into details of this, but what people who've been looking at the way many organisms develop from single cells to adults has revealed is that if you look at how early events happen and how very late events happen, there's quite a lot of divergence in strategies by which animals acquire forms. Whereas if you focus on the middle stages here, which is kind of like what I showed you in the previous picture, you could see that there is shared features regardless of what kind of animal you're looking at, right? So you could argue that you could argue that the development of each animal from a single cell follows a different trajectory and has a different final output. But there are some features that appear to be largely similar, and it's something I'll come back to again. later. I also want to point out that not all animals are like humans and fish and chick in that they're multicellular and they sort of look similar. There are also interesting animals like these two here. This is a soil amoeba called Dictyostelium, which Professor Vidyanand Nanjundaya has been studying for all his scientific life, which is in principle a unicellular organism, meaning it's a single-celled organism, but it exhibits very complex social behaviors, right? I'll show you a picture of that in a little while. So is this one here, which is a choanoflagellate, right? It's a single cell here with these protrusions here, and it swims about as a single-celled animal, but it also makes these complex rosette-like structures. And then this rosette-like structure behaves like one unit and swims around in water, often, having shifting shapes and so on. So even unicellular organisms like this soil amoeba or choanoflagellate, which is, by the way, considered the last common ancestor between multicellular and unicellular organisms, both of these exhibit very complex social behaviors. And this has sort of led to the idea that much of the machinery for making complex multicellular animals like ourselves was already in place long before we came about in the evolutionary tree, meaning that you could find their presence even in these organisms like this, right? Okay, so I'm just going to take you to, so what you will see here are these single cell animals that are sort of coming together. In time, you will see that they form an aggregate. It'll take a little while. These are all single cells. So you see the beginnings of that here. Makes a little aggregate. And then once you have a small aggregate, you have streams of cells that actually join that aggregate to form a much larger structure. But this structure actually confines itself as if someone built boundaries around it. And then it actually begins to move about like a snail, right? Not very different from how the snails or slugs you see move about. But this is the behavior of a single cell organism. Likewise, this slug will then, it's crazy. This slug will, which formed, yeah.
Here.
You have to be loud.
Yeah.
I'm hard of hearing.
Okay. So what caused the trigger of this formation of aggregate? Like they were pretty happily floating around. So why did they suddenly aggregate?
Just say that one more time.
Huh?
You say that one more time.
Why did they aggregate? What triggered the aggregation of dicties?
Yeah, I'll come to that in a little bit. I'll talk about how you build these complex morphologies. Just give me a little time, right? So they form these morphologies, and then you see once they, again, under certain environmental conditions, that will then do something like this. It will form what we call a fruiting body, right? So you see multiple of these structures forming from that aggregate of single cells formed by a unicellular animal. We'll discuss what the signals are that cause this kind of pattern formation. All I wanted to use that to say just now is that the animals that exhibit these morphologies are themselves unicellular. So you can see that there's a lot of complex pattern associated even with single-celled organisms. Okay, so that was one kind of pattern. This is a second animal. This is a fruit fly, which again you would have seen on bananas quite often.
Sorry, Maithreyi,
This
before
is
you
the
move on,
single-celled stage. Yeah.
could I ask a quick question? So we discussed when we were discussing about the multicellular organisms like human chick and all that, we talked about that there is a change in the cell numbers, there's a change in the cell types. So essentially saying there is cell divisions, growth, differentiation, and then of course also geometric complexity. But when we move to the unicellular ones, we more or less said that there is a pool of single unicellular organisms, which then exhibit some geometric complexity, right? But is it fair to say that they also exhibit differentiation of some sort?
Okay, so this is exactly the sorts of things I will get to. How do you look at how different strategies to increase numbers, to increase diversity? What are the cues that induce them, and are there different lineages? Are there single lineages, and so on? I'll come to that in a minute. Okay, so this image here is from the Drosophila embryo, and this is the single cell, or the simplest beginning of the Drosophila embryo. But at this stage, it's actually a single cell that's already divided to form a large number of cells. So the early beginnings of a fly embryo are what we call syncytial. By that I mean, okay, this movie is not playing. There's some problem with it. I'll come back to that movie in a little while. Okay, so this now is the development of a fish embryo, right? It starts off as a single cell. That single cell divides into 2, 4, 6, 2, 4, 8, 16, and so on and so forth to form a ball of cells. And you can see that all of this is happening on one side of the egg. This ball of cells then begins to sort of grow over this spherical structure. So you can see that you have cells now covering most of this sphere, and then you will see that the structure that looks like a fish is actually going to form at the edges of this sphere. And very soon you will find structures you can recognize as the head, and a structure you can recognize as the tail, and structures you can recognize as vertebrae, and so on, right? So even a fish forms by that same paradigm of having a single cell that then divides before you can get complex geometric patterns. Okay. So several people have thought about this problem of how you acquire different forms, and in fact, some of the earliest ideas were that you could explain the differences in the shapes, in this case, of many different kinds of fish, or the final shapes of many different kinds of fish, using mathematical principles. And so D'Arcy Thompson said that biological form can reflect physical and mathematical principles. And basically what he was trying to say was that a certain mathematical aspect of morphology is helpful, no, essential to the proper study and comprehension of growth and form. So the first ideas of how you can explain form were mathematical and chemical, and Alan Turing, who was a computer scientist, suggested that complex patterns of the kind you see in animals could be formed by relatively simple partial differential equations, right? And again, I'm guessing that some people will cover this during the course of this course. in between, as people began to understand how cells go about creating form, there was also a thermodynamic basis that was suggested to explain how the yellow cells and the red cells became different or sorted themselves out to distinct locations. And this is something I will come back to later on. But Steinberg and Holtfreter basically said that the complicated sorting out of cells, the creation of layers, the formation of cavities could be explained by thermodynamic principles. So it was only long after all of these theories that genes that most biologists obsess themselves with came to be identified as determinants of the spatial organization in developing embryos. And this was really work of Nusslein-Bohlhardt and Eric Wieschaus. So if you think about the ideas on pattern formation and morphogenesis, they started out being mathematical and physical, and only then invoked information contained within the genes of cells and animals as being responsible for pattern formation. As people became better able to look at what the cells within these animals were doing, you could say there was the cell biological basis of morphogenesis, which happened in the late 1990s. To just quote from Eric Wieschaus, who won the Nobel Prize for elucidating the genes are pattern forming agents, what he said was that he did not realize late in his development as a scientist that morphology and cell biology are actually the same scientific areas, or at least that the latter provides the molecular explanation for the former, right? So in other words, this era, which I would say is still ongoing, sought to ask how do cells, what properties of cells actually allow the acquisition of the complex shapes we see. And this was sort of followed by what I would like to call the return of physical biology of morphogenesis, because we started with a physical explanation for morphogenesis. And largely due to some of these people listed here, who primarily sought to understand how cells sort themselves out in embryos to build layers and that sort of thing. We now, and with the development of tools that allow us better manipulation, visualization, measurement, estimation of forces, and modeling, you could say that we're presently return to this era of the physical biology of morphogenesis. And I think it's in this context that this meeting or this course, the school, is very useful. And I would just like to point one very interesting and probably very correct statement that was made by Stuart Newman very recently, that you could look at a large variety of organisms and their development patterns by which they acquire their distinct shapes. What he said was that there must be stereotypic morphological motifs that characterize the development of diverse organisms, right? And essentially what he was referring to was that in many animals that you look at, cells will arrange themselves into tissues, right? With sometimes interior cavities, multiple layers, immiscible boundaries, containing patterned arrangements of cells. And these tissues will then elongate, fold, segment, and form appendages. What he said was that these motifs are similar to the outcomes of physical processes generic to condensed, chemically excitable viscoelastic materials. He did add that embryonic mechanisms that generate them are typically much more complex. And so he said that the origins of animal development, and you can take this to mean animal form, must lie in the mobilization of physical organizational effects that resulted when certain gene products of single-celled ancestors came to operate on the spatial scale of multicellular aggregates. And so I'll sort of try and from now on build up how single cells can do diverse things, and then try and explain how cells that are primarily unicellular organisms or from multicellular organisms organize themselves to create more complex shapes. So one of the founding fathers of physiology said that in order to try and understand form of big structures like this particular, say, heart or whatever, one must speak, so to speak, progressively dismantle the organism as one would take, as one takes to pieces a machine in order to recognize and study all its works. So that's kind of how we will go about this. And before we do that, I'd just like to give you a sense of scales here. So I will start with telling you about how a cell can acquire shape, form. But I will sort of keep going back and forth to invoke genes and proteins and lipids to explain how the cell gets its form. And in later classes, I'll take cells and try and go in this direction to say how the properties of cells can explain how organs and organisms and tissues acquire their form. Okay, so for those of you who are completely new to biology, I'd just like to say that the cell has a number of departments or ministries in it that could broadly be classified into the following. So one of the things we will speak about is where the information for what a cell does comes from. And in large measure, this information comes from the genes that collectively form what we call the genome that's situated within the nucleus of the cell. We must remember that the nucleus is not the only place that has information of this kind. The mitochondria, which are these structures here, also have their own genome, right? If we talk about energy, obviously for the cell to go about doing its tasks, it needs to generate energy. This energy comes from metabolism, which is exactly what happens when we eat food, but it's actually happening in every cell, right? And much of this energy generation, again, happens within these organelles called the mitochondria. We think about what governs the architecture of a cell. I mean, obviously, it's dependent on information that's encoded within the genes, but the components that actually enforce that architecture are primarily the membrane that sort of envelops the cell and structures that form part of what we call the cytoskeleton, right? If we look at where force can be generated inside a cell, the components that allow that to happen are essentially the same cytoskeletal filaments functioning with proteins that we call motor proteins, but acting very closely with the membrane, right? So essentially, you can think of this as interactions between the membrane and the cytoskeleton. Transport within the cell, again, relies transporting things from one place in the cell to the other, relies on cytoskeletal filaments and motors, and also membrane compartments that are within the cell. So in addition to a membrane enveloping the outside of the cell, you have compartments surrounded by membrane inside the cell that can move back and forth between this outer membrane and the inside of the cell. Where proteins are made is essentially in this structure here that's called the endoplasmic reticulum that starts off abutting the membrane of the nucleus. And then you have ribosomes, which are really the factories that take information that's encoded in the genome and convert that information into proteins. Then we have signal transduction. You can say this is communication, and this happens at the membrane. This could be the plasma membrane or the membrane of the nucleus. It happens within the cytoplasm, which is everything else other than all of these. It also happens within the nucleus. So that is the sort of cellular toolkit there is that you can play around with to alter form and function of a cell, right? And so now I'm going to take you to perhaps one of the most famous single cells that operate singly within a multicellular organism. And these are cells within our blood, right? They typically don't group together to form tissues. They live their lives as single cells within a multicellular organism. I'm going to use this movie to show you that single cells exhibit very complex behaviors. So this is a white blood cell that's chasing a bacteria that's labeled there, which it is eventually going to ingest. And you will see how it chases this bacteria, right? It pursues it for the longest time, and you will see that it keeps sort of blebbing out this part of the membrane, of its membrane, in the direction of the bacterium till it eventually engulfs it, right? And so what I would like to use this to say is that this is a single cell that in the process of either obtaining food or eating up something toxic exhibits changes in shape, polarity, and also moves, right? So as a simple way of understanding how you can build form in an animal, you could begin by asking what enables this single cell to change its shape, to become polarized, and to move, right? So we will start with that simple question. And I'm going to take a few steps back, and you could say part of this resides in genetic information, right? So I can say not all cells that are in the blood can go do this, change their shape, change their form, and move in the direction of a pathogen. So some cells are built in a particular way to be able to do this. So one question we can ask is, how do you make a blood cell that's capable of engulfing a bacteria? Or how do you make a macrophage? And at this point, we will have to invoke information present in the genes, right? And again, for those of you who may not be familiar, you can think of all the genetic material that's present within the nucleus of a cell as consisting of coding regions and non-coding regions, right? The coding regions make protein/RNA. The non-coding regions allow where within an animal those proteins or RNAs are made. And at a higher level of organization, the way in which this genetic material is looped can again control how much of that information is made and where that information is present, right? So typically, we think of gene regulatory networks. Now I'm going away from the idea of a single gene to the idea of a gene regulatory network, by which I mean you have information that's coming from some, let's say, input signals, that operates on some component of the network to affect changes in RNA or protein of some subset of genes within the genome, which is then necessary to allow changes in cell structure or behavior, right? So typically, input signals will be what we might call signaling pathways, and I'll come to this in the lectures to follow. But they can also be regulatory proteins known as transcription factors. So sometimes signals can turn on the expression of transcription factors. Transcription factors are just proteins, again made by genes, that will bind to DNA that's within the genome at specific locations and then activate the expression of other genes. Yeah? So transcription factors bind genes and make RNA, and the mRNA then synthesizes the required protein. These networks can have a fair amount of feedback incorporated into it. So if I have to make a macrophage, I need to sort of think about what genetic information is required to build a macrophage, right? Before we get into that, I just want to introduce this concept of lineage, which I think is also important. So I said one of the things that characterizes development is that not only do you have more cells as time goes by, you also have many different kinds of cells. If I look at an animal like the worm, right, C. elegans, and I think Vijay will speak about embryo of this worm during his talk, the whole adult of this animal has exactly that many cells, right? It's invariant. And what we know about each of these cells in this worm is you can trace back the ancestry of every single cell in this animal back to the single cell zygote from which it came. So this is the lineage tree, if you like, of the worm, right? Which is to say, starting from the zygote, what happened? Each horizontal bar represents a cell division. Each vertical bar represents a single cell, right? So for the worm, we can trace back the origin of every cell in the adult worm back to the founder, which is this zygote from which it came, right? So let's say we forget about a whole animal, but let's say we have a certain specific subset of cells. You can perform what you call a lineage analysis to understand every cell's ancestry back to the founding zygote. And there are now many different ways by which you can build this sort of analysis, right? It's not very different from us trying to find out our family trees, but you can do this for every cell in the animal. There are very sophisticated methodologies available. So you can say that what this allows you to do is get a single cell granularity of the molecular processes that govern differentiation, meaning that if I am a macrophage, what molecular processes allowed me to get there, and how many steps from the founding zygote did it take to get there? What molecular processes governed each step in this process? So if I actually look at a macrophage, it will look something like this, right? So let's say this is a monocyte or a macrophage, one that engulfed the bacteria. I can say that it had this founder here, this founder here, this founder here, this founder here. And we know enough now to be able to say what needs to happen to this founder to become this founder, and then to go off along this path rather than some other path and finally end up being here. So this is a sort of general question of, yeah. Yeah.
Cell division, right? And some cells reside to not undergo cell division. So I'm asking this in terms of evolutionary, in terms of
Say again?
evolution.
Say, repeat the second part.
So in terms of evolution,
In terms of evolution.
so the horizontal lineage or the ones that are dividing have more evolutionary advantage, right? Like in terms of mutations. So those cells have more chances of undergoing mutations. Like let's take mutations as an advantage in this case. So the cells which are not undergoing division will not have this evolutionary advantage. So is there a differentiation that occurs in the organism to choose the division and not the division? Like what kind of cells remain in the evolutionary aspect or in the division status, and what kind of cells tend to remain in the
What does that tell you in evolutionary?
yeah,
So
like
you can use mutations to actually map lineages, somatic mutations, right? So, okay, I've said somatic, I should just qualify. So broadly, when we talk of multicellular animals, we distinguish somatic lineages from the germ line, right? And so the germ line is essentially the gametes. So I'm not doing genetics here, but where the egg and sperm are gametes, which are the only cells that have the potency to give rise to all cells in the animal, right? And yet each of those cells can't do it by themselves. So an egg or a sperm can't give rise to a whole animal. It requires the two to fertilize to unmask that potency, right? So that's the germ line. And the somatic lineages are everything other than the germ line, and the mutations that happen in the somatic lineages are not going to be inherited in successive generations of offspring, right? But within an animal, they will be present in the cells that originated from the division of that mutated cell. So what you saw there was actually the use of mutations to try and map somatic lineages, because there, first of all, mutations are quite rare, meaning that your genome is not going to be changing by mutations very frequently. Mutations are still a rare event. So if you have a mutation in one cell, and you can track that mutation, you can actually figure out which cells came from that mutated cell. So from the point of view of evolution, somatic mutations cannot have a huge selective advantage. So whereas if you have mutations in the germline, and we will come to the germline in just a little while, we could think about what consequences such mutations would have. Was that your question?
yeah.
Okay, so the reason I told you this was that one of the other changes that I said accompanies morphogenesis is that from one cell type, you end up having many cell types. So there are two other gradients that go with this transition that we might call development. And one is that the pluripotency reduces. Okay, what do I mean by pluripotency? I simply mean the potential of a given cell to give rise to a number of different types of cells, right? So if I say an animal is made up of so many different types of cells, this is the founding zygote that can actually give rise to all the types of cells, right? But as you go along this path, the ability of any intermediate cell between here and here to give rise to all the cell types that make up the animal reduces drastically. Correspondingly, the commitment of a cell to a particular lineage, as I showed you in the previous slide, increases as this goes by, as you go on this path. So with respect to genetic information, I said making a macrophage relies on some gene regulatory network. You could ask, does this transition involve changes in genes? And if so, are these changes in the sequence of genes? Are there changes in expression of the genes? So the answer to this question is that it does not involve changes in sequence of the genes, right? So every cell type has the same genome, right? Okay, I have to raise a disclaimer and say there are a few cell types, for example, some immune cells, but not the macrophage that I was talking to you about, has bits of the genome taken out, right? But for most part, all cells retain the same genetic information as far as the sequence of the genome is concerned. What is different is what part of that information is actually translated into protein, right? So that's what distinguishes one cell type from the other. And so you could ask, is this transition reversible? And that again is an important question, because I'd like to say that, you know, my brain cells should not tomorrow become muscle cells. I'd like them to stay neurons for all of my life. And so when I ask, is this transition reversible, we're really asking how reversible is it? And if it is reversible at all, how can this be reversed? And that brings me to a second point about commitment, which is that the early dogma was that, the early thought was that when cells go from being one type to many different types, each of those types is associated with some loss of information that's contained within their genome. Hans Spemann, who's famous for something you'll hear about tomorrow, was one of the first people to show that if I look at an animal as it goes from being 1 cell to 2 cell to 4 cell to 8 cell, each of those 8 cells can become every cell in the body, right? So it's not just the founding zygote that's capable of giving rise to all cells, but every cell in the 8 cell stage embryo that can also do this, right? But he argued that really only the early divisions maintain that potency, right? But it took 3 other people to show that that wasn't exactly true, and that this potential to give rise to every cell in the embryo could also be found in later divisions. And so essentially what those 3 people were able to show was that you could take a nucleus from an intestinal cell in a tadpole of a frog and do something to it that would allow that cell to now give rise to every other cell type in the frog, which means that although it had become committed to being an intestinal cell, you could somehow reprogram it back to becoming pluripotent, right? And so essentially these sorts of experiments allowed people to argue and show that potency can be regained, but that in animals there's a check on this potency, meaning that only certain triggers, for example, serious injury, etc., might allow you to sort of make the reversible reaction favorable, right? But those are sort of under very exclusive conditions. Is the is the reversible reaction favorable in normal development. But if you play around in the lab, you can figure out what conditions make the reverse reaction favorable. Yeah, you had a question. You'll have to speak out loud.
Hello. Yeah. So in the previous slide, you had shown about the intestinal cell being put into the. So there's no fertilization in this case.
What?
There's no fertilization happening.
No fertilization.
So the role of fertilization in causing development as opposed to this process, what is the, could
So
you?
that's a good question, actually. So what you would have seen, though, if, and you probably did see it, was that these nuclei from the intestinal cell were actually put into the cytoplasm of an oocyte, from which the oocyte nucleus had been sucked out. Right? So you could say this is like the egg. This is the egg without its own nucleus. And you're bringing a nucleus from a differentiated cell and putting it into that bag. Right? So you could think of fertilization as something similar in that you're putting a sperm nucleus into a bag that has both the nucleus and the cytoplasm of an egg. So you're actually right. So what people have begun to ask, and there's some evidence in this direction, is whether there's something about the cytoplasm of this oocyte that allows this, any nucleus that's put into it to be programmable, right? So if you remember, I said neither the egg by itself nor the sperm by itself can give rise to a whole animal. You require fertilization. So what is fertilization actually doing? You could say, yeah, it makes the genome diploid, but you could argue whether all animals have two sets of chromosomes, one from each parent. But really, we think that the key thing that fertilization brings in is the interaction of something that's within the cytoplasm of the oocyte onto both the nuclei, which then become reprogrammed. And at least one aspect of that reprogramming is to unmask that latent pluripotency so that it's now become manifest.
So when you think of regeneration, is it the same process? For example, you cut off tiny pieces, and that somehow can remake all of itself. So you're really creating the environment of the zygote. So how does that come up in the regeneration?
Yeah, so I'll come to this question just a little while. So essentially, Vijay is asking whether there are parallels with regeneration and early development with respect to, let's say, the chemical environment for now. And I'll say the simple answer to that question is yes, right? But I'll come back to that question in just a little while. So, but what this actually helped establish is that this, in principle, the commitment of a cell during the course of development is not irreversible, but during development, it is unidirectional because there are checks in place to only allow it to go this way, right? But you can play around with this system. For example, as Vijay said, during regeneration, to trigger that, to unmask that latent potential that was there, right? So what this actually meant was that John Gurdon was able to take a single nucleus from a frog, from the intestine of a frog tadpole, and create a whole clone of frogs that had exactly the same genome as the frog whose intestinal cell that nucleus came from. right? So in a way, this was what we would now call the birth of cloning, and cloning got a lot of attention. But the fundamental principle that it allowed his experiments to demonstrate was that adult or differentiated cells can be equivalent to, be triggered back to being embryonic cells, right? And he called the reversibility of the state of a mature cell, for example, an intestinal cell, to one that looks like that of an embryo, meaning to one that can make all the cell types in the embryo, as nuclear reprogramming. So when we say, how do we make a particular type of cell, and we talk about lineages, we talk about the network of genes whose expression allows that unidirectional trajectory to go forward. I have lost a picture here. Anyway, never mind. You would have seen the photographs of Shinya Yamanaka and John Gurdon here. They won the Nobel Prize for the discovery that mature cells can also become pluripotent. And so since people were asking me what actually happens, what happens during regeneration, what happens when you put this nucleus. So one of the things I want to point out is that these kinds of experiments that I showed you don't always allow that nucleus to give rise to an animal. So this led to the idea that in principle this can happen, but it can happen every time you get just the right favorable conditions for it to happen. There's some stochasticity in getting those favorable conditions. And so really what Yamanaka was able to show was to take a differentiated cell from the skin, right? Let's say from your own skin, put it into a dish, add a cocktail of factors to those cells growing in that dish, and show that those cells can now become like stem cells of an embryo and give rise to all cell types, right? People began to wonder if there was a magic cocktail of factors that allowed this to happen. Actually, in principle, there are many sets of factors which in combination can do this. And so it's really about multiple ways of getting back to that embryonic state that came out from work in a dish. Okay, the other point I wanted to make was that, Arthur Waddington, Conrad Arthur Waddington, described something called canalization to explain how during development you go from having a cell that's capable of taking many different parts and giving rise to many different cell types, right? And so he sort of likened this to a ball rolling down a hill, and because of the unevenness of the surface, for example, different balls could take different trajectories and acquire different fates, right? And so essentially, this idea, so Waddington called this the epigenetic landscape for differentiation. What I just showed you was that development goes in this direction, right, of increasing commitment. And the experiments I showed you go in this direction, where you have a differentiated cell and you're bringing it back to a more pluripotent cell. And both these, both reactions are associated with very similar changes, but oppositely directed in some cases, similarly directed in other cases. But if you look at the molecular processes involved, let's say Something that gets elongated here is shortened here. Something that's expressed highly here is expressed low here, and so on and so forth. So there are certain features that increase in this direction during development and decrease in this direction during reprogramming. Processes that one kind of transition happens during development, a different kind of transition happens during reprogramming. So essentially what this allows us to see is that during development, a spatiotemporally regulated set of changes in the genome allows this to be unidirectional. And then the addition of a cocktail of factors on differentiated cells can allow us to reverse this situation, right? So this here in this lineage from one of the founding stages of mouse development is what eventually gives rise to a macrophage. And as I showed you already, that founder is somewhere here and the macrophage is somewhere there. So it's multiple steps to go from that founder to become a macrophage. Okay, so now let's say you've made a macrophage. I now want to do two other things to it. I want to try and understand how you get a macrophage to change its shape and make it move. And so here's an example of, I'll show you a movie in a minute. Here's a wound edge, a wound that's been inflicted in a tissue. And what people have seen is that when you make a wound in a tissue, these macrophages actually direct their motion and come very close or become recruited to the wound edge, right? So these macrophages had till such time been sort of moving around haphazardly in all directions. But something about wounding a tissue now causes them to reorient themselves so that they all migrate towards this wound edge. And once they've done what they need to do to the wound and the wound resolves, you will see that they will sort of withdraw themselves from the wound and take their own paths back, right? So you could say, how do you shape a macrophage? How do you move a macrophage? How do you polarize a macrophage? Okay. Hopefully this will work. Okay, so in this movie here,
Maithreyi,
one of the most, yeah.
in the previous,
Watch this movie, ask me that question. So this is one of the most sophisticated microscopes available on the planet today that allows you to look at cell organization and dynamics at very good subcellular resolution. And what you saw, you will see that's, come back in a little bit. You can see many different things, but just play this thing again. So that thing there is the macrophage. You see how quickly it's moving about, how quickly it's changing its shape. Everything that's inside it is stuff that it has engulfed, right? So this is in the inner ear of a zebrafish embryo, right? So what you can see is that, so what you saw in that first movie that I showed you was a macrophage imaged by video microscopy many years ago in the 1950s, where you saw it in a sort of 2D way, now you can sort of imagine the three-dimensional features of this macrophage migration, right? And so I think you can see how, at what time scales, the shapes are dynamically changing. And this cartoon here is marking the trajectories of a macrophage. Okay, sorry. So if you look, so if you look at the trajectories of this macrophage, prior to, let's say these are several macrophages, prior to it being wounded and after having a wound, you will see that there are many features about the way this macrophage moves that are influenced by the process of wounding, right? And you can see people have quantified many different sorts of indices, and this one here is called the meandering index, which, as its name suggests, tells you something about how persistently a macrophage follows a particular path. So let's just say that you have some source of information, such as a wound or a bacteria, like we saw in the first slide, and something about this source is allowing this macrophage to modify its motility in such a way that it can efficiently approach that source, either to engulf a bacteria or to heal a wound. And so again, I think Vijay will try and cover aspects of random walks and that sort of thing in his classes, but I just want to say that what appears quite clear is that the source, which may be a wound or a bacterium, can influence the trajectory a macrophage takes. So as biologists, you could ask, what is it that a bacterium or a wound provides that allows a macrophage to exhibit very complex changes in shape, 2, acquire polarity, and 3, move in a persistent manner. Yeah.
So what is that image supposed to represent? The one at the top left?
Say again?
What's the image at the top left?
What's in the top?
Yeah.
Okay, the top is just some drunken guy who's trying to reach home after a night out at the pub, right? He will get home. He will probably get home, depending on his state of inebriation. But how he reaches home or what track he follows may depend on what happened at the pub. Okay, so in a way, I'm just okay. So anyway, that was basically just to illustrate a random.
Maithreyi.
Okay, so now what?
Maithreyi.
Yeah.
Yeah. Coming back to my question. So this is about the first video that you showed about the macrophage following the bacteria. So there the macrophage has like a biased, persistent or non-persistent movement, but the bacteria also seem to be like running away from the macrophage.
Bacterium also?
Seems to be running away from the macrophage. So in a sense, is the bacteria also polarized and has a non-biased movement, or is it just like randomly moving around?
Okay, so in the lab, we can perform controlled experiments. So let's say you can crush up bacteria or some other chemical, fill it up into a pipette tip, and simulate the effect of a moving bacterium by sort of creating your own trails, right? So studies of that kind. So I'm not going to treat this as a design in sensory whatever, but I'll come to that point with more easily interpretable studies in a dish. Yeah, and I will do that in the context of cells that have to migrate within an animal to actually allow it to acquire its shape. I'll tell you there what we know about the shape of the gradients, how those gradients originating, how long-lived those gradients are, and that sort of thing. I'll do it in that example if you don't mind. Okay, so I just want to use this opportunity to then tell you how can any cell change shape or become polarized or move. So if you want to influence cell behavior, you need to be able to have a toolkit that allows a cell to do that. I'll try and show you that really the toolkit that's responsible for this in a cell is the link between the outer membrane of the cell and the cytoskeleton. So the membranes of cells are typically phospholipid bilayers that are not homogeneous in their composition. They have differences in the proportion of different kinds of lipids. They're also different across the membrane with respect to proteins that are embedded within the membrane. So if you think of composition, you have to worry about both the protein composition and the lipid composition of the membrane. And these are important also for conferring physical properties to the membrane, including the rigidity of the membrane and its curvature. The nanoscale organization, by that I mean this mosaicism of the formation of clusters of proteins within the membrane that organize themselves into domains, allows proteins to exert some action very locally, right? This could be signaling, and again we will see in another example how this can happen. Membranes also have specific classes of proteins that allow cells to adhere to and exert traction forces on a substrate on which they move. So remember, although you saw this cell sort of appearing to swim in the sky in the movies I showed you, they're actually not, I mean, they're actually stuck onto something, even if it is a matrix of other proteins, right? And so this allows cells to adhere to and exert traction forces on a substrate in the same way that we walk, right? And we also have in multicellular animals proteins that allow cells to modulate how strongly they're stuck to their neighbors, right? And so these molecules allow the membrane on which they're present to also link to the cytoskeleton and contribute to the amount of tension at the interfaces between adjacent cells. Likewise, the cytoskeleton is made up of polymers that also self-assemble and whose network organization depends on interacting proteins. And their ability to generate force depends on polymerization against or anchoring to the membrane or the action of motor proteins. I'll take you through this in a little detail, but not too much. So typically, we speak of two sets of cytoskeletal elements, the actomyosin system and the microtubule motor protein system. And while both of them are involved in generating force, the actomyosin system typically allows you to generate tension. Microtubule motor proteins can be thought of as load bearers that help resist compression. And so just a textbook view of the membrane is like this. You have a lipid bilayer. Each of these rectangles is a layer of lipids. These lipids are arranged in such a way that the heads and the tails of the lipids are oriented such that the heads are away from each other and the tails sort of at the middle of this planar bilayer, right? Typically, we think of this plasma membrane as a fluid mosaic membrane. And the fluidic nature of membranes is because of the rotational and motion properties of lipids and proteins. The lipids themselves provide the basic structure of the membranes. The proteins carry out a vast array of specialized functions. When we talk about membranes, we should also talk about membrane reservoirs, because this is important when you think about being able to change cell shape. And these are essentially membrane buffers that help redistribute the area of the membrane when a cell needs to stretch or change shape and size, right? And in a simple way of looking at it is to say that, you know, the membranes can have lots of infoldings within them that kind of get stretched out and increase the surface area available when, for example, they need to increase in volume. So you could, some of the contributors to membrane reservoir are these membrane superstructures that could be either large membrane folds or small invaginations, or compartments that are membrane compartments that are inside the cell. All of these could help increase the area of the membrane when necessary. The other thing you should remember about the membrane are the sorts of functions that it typically performs. And one of the functions is that it acts as a barrier and or an interface between the exterior of the cell and the interior of the cell. But, you know, it also allows communication between the exterior and the interior. Can be thought of as a hub for biochemistry that happens inside the cell. It allows, it's a hub or a location within the cell that allows the cell to modulate its mechanical properties. And it also acts as a way by which you can compartmentalize biochemistry, either on the large surface of the plasma membrane or in small compartments inside the cell. And the composition of both lipids and proteins on the membrane can influence not just curvature, but also things like rigidity, like I already said. And I said that a special class of molecules that allows cell membranes to be mechanical hubs in the cell are adhesion molecules that either link the cell to the substrate, as you see here, or one cell to the other, as you can see here. And they act as mechanical hubs because both of these classes of adhesion molecules allow you to form a link between the membrane and the cytoskeleton. So in some ways, when we discuss polarity, motility, and shape, our focus will be on looking at how you can modulate this membrane-cytoskeleton link to allow changes in morphology to happen. So one more class of, or one more set of, one part of the toolkit that we should also remember are the various membrane compartments themselves. And as I already said, you can see that these are the superstructures of the membrane that I was talking about. And they're constantly exchanged between the plasma membrane and the inside the cell. So in addition to carrying, or in addition to being able to compartmentalize biochemistry within these compartments also allow you to modulate the amount of membrane that a cell has. Okay, I see that I've run out of time. How much longer can I go, Vijay? Can I try and take 5 minutes and finish this part? The other feature of the membrane is that I think I forgot to say that the membrane by itself is inelastic. It cannot, you can't simply mold it into any shape you want. So if you actually have to change the shape of a membrane, something active needs to happen. One of the things that can modulate the shape of a membrane are the shapes of the lipids that are inserted into the membrane, and whether you have conical lipids or inverted conical lipids, you can achieve opposite effects on membrane curvature. Likewise, you can have the shapes of proteins that are inserted into membranes influence the curvature of the membrane. And then the action of the cytoskeleton and the ways by which the cytoskeleton polymerizes can also modulate on a larger scale how the membrane is shaped. Yes, let me just. Okay, so what I'm going to do then is, shall I move to the cytoskeleton later? Yeah, I think we will stop here. So I think what I've told you is what properties of the membrane can allow a cell to change the amount of membrane it has, which can allow it to shrink or grow, change the curvature of it. And what I'm proposing to do next is to discuss how you can modulate the assembly of the cytoskeleton, and then how you can use the two together to explain how a macrophage changes its shape, polarizes, and moves. And after that, we will go to tissues, how cells make tissue.
Questions? The mic in your room.
I must have missed something. I'm wondering what is the relationship between the irreversibility or unidirectional processes and reducing potency or maintaining the same potency? Because in cell division and I think cell differentiation, both are irreversible. I don't know, right? Or
can someone repeat that question?
I mean, cell differentiation and cell division are both irreversible. Am I right? In terms of biology, you call it both the cell differentiation and cell division as irreversible processes, right?
Then I'll shout the answer out loud. Yeah, tell me.
I mean, in biology, in cell division and cell differentiation are called irreversible processes, right?
No, so I said cell differentiation during development, under normal developmental conditions, differentiation is irreversible. But if you have some stress, for example, injury, right, you can do something you can call trans-differentiation, meaning where a differentiated cell can either become a different kind of differentiated cell, or it can go back and become a stem cell and then give rise to new cells that are required to heal the injury.
My question.
Cell division, you said cell division also. Cell division is irreversible in the sense that once a cell has committed to dividing, it will make two daughter cells. unless, of course, there's some defective protein that arrests this division at some step in the process. So, for example, you could sort of separate the chromosomes to the two daughter cells, but not be able to physically constrict the region in the middle so that you form two daughter cells. So that although you divided the nuclear material into two, it still exists within the same cell.
So,
but when you say cell division is reversible or irreversible, we speak of cells that are mitotically active. By that I mean they are capable of dividing. So one cell becomes two daughter cells. Each of those daughter cells can then go on to divide and give rise to more daughter cells. But there are also cell types that where the ability to divide is lost, right?
Yeah.
Meaning that once you've made a differentiated cell, you can't create more differentiated cells by dividing that cell, right? So to make, let's say, new neurons in my brain, I can't divide a cell that's already a neuron to become two neurons. I have to go back to a stem cell and differentiate it to become new neurons. Whereas for some other cells, like for example our skin, this division constantly happening. Of course, that's also happening in subsets of cells. But broadly, you can say there are some cells that are capable of dividing, and each daughter cell is capable of dividing to give rise to more cells, and other cells that have lost this ability to divide. So if you want to increase their number, you have to go back to the stem cell from which it came, and then initiate the program of differentiation, right? So for example, if you have injury in muscle, there are some specific cells that are called satellite cells that are normally arrested in the cell cycle. But in response to injury, you can reinitiate division and differentiation in those cells. So you make new cells that will now go and become part of muscle.
If we make sure that a process is irreversible, it doesn't mean that it's reducing potency. Yeah,
it's reducing
the potency. It is becoming differentiated or not, because if I'm understand correctly, in both conditions, in cell divisions and cell differentiations, they are unidirectional, but in some cases we have the reducing the potency, but in some cases we don't. I just wanted to make sure that I'm understanding correctly or not.
Okay, so I think what I meant to say is that in principle, every differentiated cell is capable of being reversed back to becoming a pluripotent cell, right? During the course of normal development, this does not happen. It doesn't sort of keep going back and forth, but some trigger, there needs to be some additional trigger to favor the reverse reaction. For example, injury.
I see.
Right? I said that to say no cells in my brain, no neurons in my brain are becoming muscle. Right? Yeah, we could use that metaphorically to explain, sort of say why I've become dumber over time. But in principle, there's also stability of the differentiated state. That's what I want to say. So during development, there's stability of the differentiated state, but a stability that can be potentially reversed and taken back to the pluripotent state. This does not normally happen, but will happen when there's some need or some other trigger. Yeah. So Vijay was talking about regeneration. There are some animals where regeneration can happen very effectively, meaning If you look at planarians, right, you can chop their heads off and they'll grow a new head, right? Or you can chop their tails off and they'll grow a new head. Lizards will grow a new tail, right? But if I chop my finger off, I'm not going to grow a new finger, right? I'll have an amputated hand. So the question is, does regeneration also, I mean, what Vijay was asking is whether regeneration creates the, let's say, activates the same network of genes that normally used to reverse differentiation in a dish or these special cases.
Other questions?
Ma'am, as you saying that I have a doubt, means some injuries happen, then hello, yeah. So some injuries happen, then only the some cells are differentiated again. So as I know that every organ has some very low number of stem cells. Are these stem cells or somatic cells? That is my doubt.
Okay, so it's true that every organ or every cell type, or many cell types have what you might call stem cell precursors within the organ. These stem cell precursors are not typically pluripotent. So if I say I want to make more macrophages, I won't go back to a founder that looks like the zygote, right? But I'll go back to a founder, I meaning I'm not going to go back, the animal goes back to a founder that can make all different types of blood cells, or a stem cell that will give rise to a few lineages of blood cells. So you're right in saying that you have stem cells of different capacities, right? So these aren't pluripotent. They could be more, they could be, when I say pluripotent, I mean giving rise to every cell type in the embryo. When I say multipotent, I mean a subset of types. For example, all types of blood cells, right? So it's true that for many tissues, there are sort of stem cell progenitors that can give rise to one or more lineages of cells that make up this tissue. Again, if we look at neurons in the nervous system, for a long time it was a debate about whether or not you could increase the number of neurons, right? And again, work in this direction is still controversial, but people have identified regions in the brain where you have some stem cell progenitors that can make some types of neurons. They're not there in every part of the brain. They're there at some times in the life of an animal, right? So I think the understanding, I'm not a neurobiologist, but the understanding is that there are certain regions of the brain where you can use some stem cells that are available to produce new neurons, but you can't do that in all regions of the brain. So in some regions of the brain, if you lose neurons, that's it. That's as many neurons as you've got for your lifetime. Yeah?
Maithreyi, so in case of cell divisions, so most cells have a limit to how many times they can divide, after which they generally reach senescence. So when this nuclear reprogramming happens, does this limit also hits reset, or is that maintained?
Okay, people have spent a lot of time looking at reprogrammed cells, partly because you can make whole animals from reprogrammed cells. People have asked, are reprogrammed cells as good as conventional pluripotent cells? So one aspect they have looked at is senescence, and in particular, they've looked at telomere length say, for example, in Dolly the sheep, which, by the way, was the first animal to be derived from the nucleus of a differentiated cell. And one of the things they did see was some signatures during young age of Dolly that suggested that senescence probably progressed faster. And this is, yeah, people are looking at that a lot.
Okay, so let's take a coffee break and come back around 4:10.